Alrighty, so this is the notes for Unit 3, and it is chapters 8, 9, and 10 in the book. It is metabolism, so energy and enzymes, and then cellular respiration and photosynthesis. We'll break it down into little chunks of about 11 slides per synchronous day, so hopefully it'll be a little bit manageable. I also am going to be doing labs every day to sort of give you another look at the content. So, sorry. Okay, let's go. And so metabolism is all of sort of our chemical reactions in our body. So how quickly we break down food, how if we store food, so like some people, they might say you have a really high metabolism or you have a slow metabolism. It's just sort of all the enzymatic chemical reactions in our body and how that kind of comes together to burn or maybe not burn energy, and then give us sort of sometimes some of our traits. So a metabolic pathway is basically a chemical reaction catalyzed by a biological enzyme. And so when we have something and we break it down into something else, that's a chemical reaction. And so when it is one of those, when it has to do with metabolism, we just refer to it as a metabolic pathway. Almost every chemical reaction in our body is metabolized by a very specific enzyme. So every process has its own special little enzyme. And so, for example, like lactose intolerance, if you eat like a bowl of ice cream and it has lactose in it and you are lacking the enzyme lactase, then you can't break it down, and then you get a bellyache, and then you don't feel very good. And so that lactase and lactose is like a little puzzle piece. They fit together, and it's very specific. So you might be lactose intolerant, but you're not like fructose intolerant. So you could eat like other sugars because you have fructase as the sort of corresponding enzyme. And so this is just an example of sort of a series of reactions, and each little separate reaction, or each little from like A to B, B to C, C to D, is catalyzed by its own little enzyme. And so a catabolic pathway is one that catabolism is, or when something is catabolic, that means that something is broken. If you had me for chemistry, you might remember that we talked about anabolic and catabolic reactions. And catabolic, I said, because cats are kind of like jerks, and so they break stuff, and so they're sort of like, I'm going to push that vase off the shelf, and so a cat breaks stuff, and so catabolic reactions break things apart. And so in chemistry, if you remember, when we have a bond and that bond is broken, then energy is released. And so in a catabolic reaction, energy is given off. And so the example that we'll talk about in this unit a lot, along with some other stuff, but the big one is cellular respiration. So we have glucose, and we stored the energy that we captured from the sun into bonds of glucose, and then when we go through digestion, we break those apart. So cellular respiration is a catabolic pathway. And then anabolic is the opposite. And so it's the, hang on, my lights just flipped off. I haven't moved in long enough, so, okay, sorry. And so anabolic is the opposite. So if catabolic is breaking, then anabolic is two things and we're putting them together. And so if we make a bond in chemistry, we are storing energy. And so photosynthesis is an anabolic pathway because we're taking the energy from the sun and we are making a bond and storing it into the bonds of glucose. And then energy, just like if you took physics, or if you sort of think about it, it's on here it says the capacity to cause change. It's the ability to do work. And so if we have energy, we can do things, and sort of a generalized, but if you sort of think of it in the lens of biology, it's so that we can do chemical reactions. And then different kinds of energy, and some of these you probably know, and some of them we'll just think about a little bit. Kinetic energy is stuff that's moving, and so that's just one that you probably know, like if you roll the ball downhill, then it has kinetic energy. If it's sitting at the top of the hill, it's potential energy. And so then heat is, when we refer to that, and you probably learned this in physics, maybe chemistry, that's thermal energy. And so we give off a lot of thermal, we give off a lot of heat, we lose a lot of energy through heat, and sort of the loss, sort of like we feel warmer and stuff like that. And then potential is that because it can do something, sort of the ball sitting up at the top of the hill, and then it sort of goes down. And then the potential energy that we care about with our bodies is that contained within chemical bonds. And so chemical energy is potential energy in bond form. So it's sort of, it's available and it can do something, but it just sort of sits there until it's time to. And then we can convert it to different things. We take light energy and we convert it into chemical energy. We take chemical energy and we convert it into a different form of chemical energy, or maybe use it to do the processes in our body. And so then we're going to talk about the two of the laws of thermodynamics. And so it's just how we take energy in and out, and sort of how it sort of goes through our system, and then how it gets transformed and used or lost or different stuff like that. So in a closed system, it is controlled, just like this. The example on this one is a thermos. So it holds it. It's all sort of contained within a system. Living things aren't closed systems. We are open systems, so is the universe, and so it just sort of, we release it and it goes. And so the big one that where it goes is heat, and so that heat then has sort of a cascade effect on the chaos in the world or in the universe, which is entropy, and we'll talk about that in a bit. And so the first law of thermodynamics is we can't destroy it or create it out of nothing. And so it goes from one form to another. And so the energy from the sun is nuclear fusion, and then whenever that fuses, that releases energy. That energy comes down as photons. We capture the energy from the photons through photosynthesis into the bonds of glucose. We take the bonds of glucose, we eat it, we digest it, we break the bonds in the glucose, we turn it into ATP, and then we lose a little bit as energy, or a little bit as heat. So it's this big cycle of things sort of lost, but it doesn't really, it's not like it goes poof out into the atmosphere. It's that release of heat as it comes out of our body increases the chaos of the universe, and then we talk about expansion of the universe and sort of how the universe is constantly getting bigger, and a whole bunch of stuff that doesn't really have to do with this class, but is super cool and interesting. But if you're interested in like astronomy or stuff like that, then that might make sense to you. The second one is has to do with entropy. And so every energy transfer or transformation increases the entropy of the universe. And so we are constantly increasing our disorder. One of my favorite movies is Jurassic Park, the first one, and Jeff Goldblum is in it, and he like puts like water on his hand, and he's like, well, all the imperfections in your skin and the water might go this way or it might go this way, and it's disorder and everything is chaos. This is what he was talking about. And so that energy that we lost as heat actually goes and contributes to the disorder, or it increases the entropy of the universe. Deep science thoughts. And then this is just talking a little bit more about that. And so whenever we make an organism, whether we're making glucose, whether we're making skin, or whether we're making like a person, that is forming something, and that decreases entropy because it increases order, sort of the organization of a system. And so that sort of localized organization doesn't really counteract the overall disorder in the universe. So sometimes the people talk about, well, it doesn't make sense because it's against the natural laws of entropy. Then why should things become more orderly? Because that's the way evolution works. We became more complex because that's just how survival of the fittest. We'll talk about natural selection in the spring. And so then we talk about a thing, and sort of we'll talk about it's how we measure how disordered or ordered a system is. And so we'll talk a little bit about free energy. And so a living system's free energy, so it's sort of, think of like a area or an organism or chemical reactions within our body. So that free energy is the energy that can do work when temperature and pressure are uniform. So we have a given temperature-ish. We have, I would hope, a given pressure. That would be weird if we were like overpressured or underpressured. And so when we have sort of a controlled system, we can measure and predict things with using free energy, which is up at the top of the slide. The symbol is delta G. And so the reason it's called delta G is because it's called Gibbs free energy. And so the big capital G comes from the first, it's the last name of the guy that came up with this. His last name's Gibbs. And so the change in free energy, so delta G, is during a process is related to the change in enthalpy, or the change in total energy, change in entropy, and temperature in Kelvin. And so all that stuff might like go in your brain, but we're going to do some practice, and so it'll get a little bit better as we go through. And so delta G equals the change in total energy minus the entropy, change in entropy, multiplied by temperature in Kelvin. So this will be something we're going to do some math problems. Might be some grid-ins, so something that maybe when you're studying you should pay attention to. And then if we get a negative delta G, those reactions are spontaneous. And so spontaneous means it just happens. And so if it just happens, that means that we're probably increasing the disorder of the system. If we are increasing the disorder of the system, bonds are being broken. If bonds are being broken, energy is given off. And so then we can measure the energy within a system, and if it decreases, we know it's a spontaneous reaction, and we can predict our Gibbs free energy value will be a negative number. So let me say it again. If we say this reaction is spontaneous, that means it just happens. And stuff that just happens is stuff that increases entropy. And so it increases entropy. Remember, that was the disorder of the system. And if you break a bond, you're increasing entropy, and you're also releasing energy, which then we can measure oftentimes as a change in temperature. And so this just talks about, sorry, the sort of the stuff that I was just talking about. And then it talks about equilibrium. The whole goal of our metabolism is equilibrium. So we want to get to a steady state, whether it's temperature, whether it's digesting things, and so we use the particles and the rest get eliminated as waste. That's sort of the goal of the whole thing. So we sort of, we have maybe a bunch of change and then we sort of level it out. And so this little graphic, if we just look at it a little bit closer, it says more free energy, less stable, greater work capacity. Now, if you take a second and think about those should make sense that they go together. If there's more stuff out there, there's more energy, it's less stable, and then there's more stuff to do the work. And then if we sort of go to the next one, it says, in spontaneous change, the free energy of the system decreases. We go from orderly to disorderly. So then the energy decreases. We approach an equilibrium. And so we were approaching stability. And then release free energy can be harnessed to do work. We'll talk about an example in a bit. And then we get to the point where we have less free energy, we're more stable, but we have less capacity to do work. Less energy, less work, more energy, more work. So hopefully conceptually that makes sense. And then we have kinds of reactions. And so this is just vocabulary. And the nice thing is these words sound exactly like what they mean. Exer means given off. Ender means put together or sort of brought in. And so exergonic proceeds with a net release of free energy and is spontaneous. And so all spontaneous reactions are exergonic. They have a negative delta G. And so then on the opposite side is endergonic, is the stuff is becoming more orderly. And so energy is going into the system. We're becoming more stable. We're becoming, we're sort of going against the grain of letting stuff go. We're sort of going against that. So then these would be non-spontaneous. These would be a positive delta G. And so then if we look at these for a minute, if we look at the top one and we didn't know what kind of a reaction it would be, we'd have a lot of energy and then we'd end up with less energy, which means energy was given off, which means that is exergonic. We could also say that is spontaneous. And then if you also look at this, we have a decrease in delta G. So we would see that if we did a equation, it would have a negative answer. And then the bottom one is the opposite. We have a little bit of energy. We increase the energy in the system. And so we are bringing it in and we're storing it. And then that means that that is a non-spontaneous reaction or an endergonic reaction. So we would put it in, and this is when we store stuff in bonds. And so oftentimes in our bodies, we do all kinds of work. So we have chemical work, we have transport work, we have mechanical work, so we build stuff, we move stuff, we enzymatically react stuff. And a lot of times our reactions are what's called coupled. We have to spend a little bit of money to make a bunch of money. And so we have to spend some money in order to get more. And so we have to use a little ATP to make a lot of ATP is one of the things. I'll show you another example in a second. And so the big sort of currency, the dollars, the money of our body that we sort of pay things, the currency is ATP. So it's adenosine triphosphate. And so it has a sugar, a nitrogenous base, and 3 phosphate groups. And so if you remember other stuff that we talked about way back at the beginning, we had a sugar, a phosphate, and a nitrogenous base. So these are very closely related to nucleotides. And so we just have a picture. And so if we have this, we have ATP. I'm going to flip back. Oh, nope, I can't. It won't let me. And so we have an adenosine and the triphosphates. And so there's 3 phosphates. The one between the second and the third phosphate is the bond. That's sort of the big bond that holds all the energy. And so we can make ADP, diphosphate, and then an inorganic phosphate. We can also make AMP, adenosine monophosphate. It doesn't happen very much. It takes a lot more energy to break that bond, and so it's not really worth it. But it is a thing, and we'll talk about it in the next unit. And so this is just showing how we can break that, what's called the terminal bond, the end bond of the ATP, and then we end up with ADP, and then inorganic phosphate. And since we broke a bond, we have some energy now. And so we use that to do the work in our bodies. And so we talked a little bit about the 3 kinds of things, the mechanical, transport, and chemical. And so remember at the beginning, we have hydrolysis. And so lysis means to break. Hydrolysis is how we break stuff apart in our bodies. And so we have hydrolysis to break off that third phosphate from our ATP. And so then this is an example we're going to look at a little bit closer. That is a coupled reaction. And I don't know if I can move me around or not. I'm going to turn me off for a second. And so on here, it shows us that we have glutamic acid and ammonia. And so if we put those 2 things together, it becomes more orderly. And so that's an endergonic reaction. And the delta G is a positive number, which we talked about before. And so if we look at where it says number 1, it says ATP phosphorylates, which means it puts a phosphate onto glutamic acid. And so we would have a phosphate on that little green triangle. And then it makes the amino acid less stable. Glutamic acid likes the way it is, and when you mess with it, it kind of gets like zingy, and it doesn't really like it, and so it doesn't really want to stay like that. And so then we have ammonia. For number 2, it says ammonia displaces the phosphate group, and then it forms what's called glutamine, which is the same thing that we had up at the top. And so it pops off that phosphate, and so then now we have the glutamine plus the inorganic phosphate. And then it says coupled with ATP hydrolysis, so which means that we broke ATP apart using water. And so when we broke that apart, that was exergonic, because when you break that bond, it releases it. And so if we take the delta G's from those two things, so our top one gave us, it was a positive 3.4 kilocalories per mole, and then our hydrolysis of ATP gives us negative exergonic, negative 7.3 kilocalories per mole. We have a net of negative 3.9 kilocalories per mole. So while the first reaction, the glutamine reaction, sort of absorbed or took up some energy, overall, we had to do that in order to get our ATP broken down into ADP and inorganic phosphate. And so overall, this was being energy producing coupled reaction. Hopefully that makes sense. And there's my face again. Alrighty, and so when one of the big things that we'll talk about is our body likes to shift around the phosphate groups. And so whenever you pop a phosphate onto something, you are phosphorylating it. So it's exactly what it sounds like. And then this just talks about how if we take ATP and we break the bond, and we have ADP and inorganic phosphate, and then we form the bond, and so then we store the energy, and then we break the bond, and then we use the energy, and then we go over and over and over and over and over. So that's what it's talking about when it says that it is renewable, or maybe regenerated is a word that you might see. And so this just shows that in a graphic form. And then one of the things in our body that we have to sort of overcome or lower or make a little bit better is activation energy, and that's where enzymes help us. So if we go back to the example about lactose and lactase, we will eventually digest that ice cream, but it is so uncomfortable that then we get an upset stomach, and then other bad things might happen, and so it happens too slowly, and so we sort of want to speed up that sort of reaction to break that lactose down. And so we have to sort of get over a little hump of what's called activation energy. It's if you might have learned about this in chemistry as well. And so we have to get over that, and one of the things that helps us get over that is enzymes. And so this is just a graphic that shows us that if we want to take our reactants and turn them into products, we have to get up over sort of that hump of energy. And so this is what I talked about. The enzymes lower that energy. They lower that sort of hump that we have to get over. Now, it does not affect the change in delta, or the change in free energy. It does not, like we're starting here and we're ending here, and we might have to, without the enzyme, we might have to go all the way like that and go down. But with an enzyme, we might just have to go like that and go down. The sort of difference between where we started and where we ended does not change. Just that sort of hump that we have to get over is decreased when we use an enzyme. And so this just shows a graphic of sort of the starting and the ending is the same. So the delta G is affected. Please remember that. But that activation energy is decreased with enzymes. If we don't have the enzyme, like we don't have lactase, we'll still get to that end product. It might just take a while and be super uncomfortable. And so then people are smart and they're like, oh, I won't eat milk. Or they were like, oh, or maybe I'll take some Lactaid. So then they're happy and they can have their ice cream as well. And so this is just a little bit of chemistry vocab. So we have enzymes, we have substrates, we have an enzyme substrate complex, they fit together. The active site is sort of where it fits in there. They fit like a little glove or like a little puzzle piece. Induced fit is when I put my two pieces together, and then when they sort of get nice and sort of comfortable, it makes it so that that little environment is so perfect for my enzymes to work, but they have to be together and they have to fit. And so this just gives us a picture of that. And then there's a few things that affect enzyme activity. A big one is temperature. We talked about this when we talked about the pineapples, and I cooked the pineapples. We had fresh pineapples, and that was denatured is the word that we used. pH also does that. We'll do a little lab about potatoes, and we'll sort of investigate a little bit more about pH and temperature and concentration of enzyme. And then, so this is just talking about the pH. We have an optimum pH in our body. It's why if we get too acidic or too basic or too dehydrated or things like that, our body starts to not work very well. If we get a fever and we get too hot, it can denature our enzymes, which is a huge problem. And so if you've ever gotten a really high fever, you probably had to go to the hospital, or like my mom when I was little put me in a freezing cold bath of water, and it was terrible. But hey, I didn't denature my enzymes, so thanks, Mom. And so then we have these little things that sort of are little helpers, which we have every once in a while. And so we have two little helper molecules that help our enzymes. And so they're called co-somethings. And so we have cofactors are non-protein enzymes. And then they are also usually inorganic. So like this says on here, metals or ionic form. So this might be like we need iron in our diet. And so we need different calcium, stuff like that in our diet. And so they're necessary. They're not really organic, but they're necessary to help us, so sort of the enzymatic things in our body work properly. Sort of very closely similar to it is a coenzyme. And so this one's just one that is organic. And so it just, it contains carbon. So like if we need like nutrients and like vitamin B and different stuff like that, because those help our enzymatic processes as well. So it's just two little categories, but they're kind of the same. And then inhibition is exactly what it sounds. So if it's competitive inhibition, it means that here's my enzyme and substrate and they sort of fit together, and then something else comes in and they're like, we're going to take the spot and we're going to compete with that spot. And so then it sort of takes up that space. And so that may happen, or we might have our the enzyme and we have something that sort of squishes down here and then it sort of squashes the shape of it. And so this would be a let me get my hands in the right spot. This would be a competitive inhibitor, and this would be a non-competitive inhibitor that sort of makes it impossible for the enzyme substrate complex to work, but it's not really in that spot competing. And so this is just showing that graphically. And then feedback inhibition is, a lot of it is how our body maintains homeostasis or equilibrium. And so we'll make stuff if we need it. When we get too much, we stop. And so it's sort of like your thermostat at your house turns on, cools your house down, gets cold enough, shuts off. Then it starts to get warm because the sun's out, kicks back on, kind of like that. And so then we have this picture that if you look down at the bottom, the end product of the series of different reactions is isoleucine. And so if we sort of follow the little red arrow up to the left, that isoleucine becomes a non-competitive inhibitor for that the purple little enzyme there. And so if they make a bunch, it sort of gets jammed up on there, and then the factory shuts down. And then when we need it, and it sort of, then the one gets released and it goes and it gets used by the body, then we're like, oh, we need some more. And so when that non-competitive inhibitor is gone because it had to get used, then it sort of opens it back up, and then the whole little system turns back on. Alrighty, so we're going to talk about cellular respiration, which is the process by which we use glucose and turn it into ATP. And so this is just talking about how energy ultimately comes from the sun, and then through photosynthesis we put it into glucose, and then this is the process that we take that glucose and then turn it into ATP. And so just a little figure that goes along with it, just same sort of thing. And then I think I've talked about before, but just in case I haven't, ATP is sort of like the currency, the money that our body spends. And so we have to turn it into money before we can do some work. And so there's a couple parts of when you break down glucose. Some of it can be done outside of the mitochondria, and some of it is done inside the mitochondria. And so fermentation is anaerobic and can be done in some environments and also by certain organisms. And then aerobic is it needs oxygen, so that's what, and oxygen is present inside the mitochondria. And so cellular respiration is taking glucose, which we eat, oxygen that we breathe in, and then changing it into a product, so ATP, and then a little bit of water, and then carbon dioxide, which then we breathe out. And so we are can do, like I said, we can do some anaerobic, but really efficiently sort of high power stuff is we have to use oxygen. And so whenever we talk about these, this is a little bit of stuff from chemistry, but just a refresher of redox reactions. And so sometimes things are oxidized and sometimes things are reduced. And these words can be hard to understand. And so the way that I remember them is reduction means less. That's just what the word means. And so when we're talking about moving electrons around, electrons are negative. And so if I gain negativity, I am less than. And so when something is reduced, then it gains electrons. And then the opposite of that is oxidation. And so if something loses electrons, then it is oxidized. And so on this, if we just look at just some examples of it, is we have the sodium and the chlorine, and then we talk about sodium chloride. And so this is like ionic bonds 101. And so when the sodium loses the electron, it's getting rid of negativity, so it becomes more positive. So it's the one that's oxidized. And then the chlorine is the other one. And so chlorine gains that electron, it becomes more negative, it is reduced. And so on here, with the little table at the bottom, one of the things that we talk about is hydrogen, and hydrogen is special. And so hydrogen can be positive, or we can talk about electrons that are negative, and so it's kind of a weird thing. So I'll talk about hydrogen, which you've always learned is H+, but then it sort of adds to different things, a positive thing to make a neutral thing. So it can kind of work as an electron, kind of work as a proton, positive or negative, because hydrogen is different, it's special. And so this is just some vocabulary that does with it. So the thing that does the reducing, so the thing that sort of causes that thing to get reduced, so that thing over there got reduced, I did that, and so I was the reducing agent, something like that. You understand what I'm saying? And so then if I take away that negativity, then I'm becoming more negative, and so I'm allowing that thing over there to get oxidized. So I'm the oxidizing agent. And then just a little bit of extra information underneath there. And so when we talk about cellular respiration, we have these little special little molecules. And so if you've ever been to like the Pepsi Center and they have the little guy with the yellow cart and he like grabs you by the Pepsi Center, he's like, I'll drive you to your car, and then he takes you down to your car and he dumps you off down there, and then he comes back and he gets another person, he takes them back and he dumps them off. That's how I sort of envision the coenzymes that work in cellular respiration and photosynthesis. The first one we're going to talk about is called NAD+, and so it's sort of a charged up thing that's looking for something to come along and make it stable. And so if you look down a little bit, it says that the NAD+ acts as an oxidizing agent during cellular respiration. So that is that NAD+ allows that thing to get oxidized, so it is letting that thing become more positive, so it's taking an electron, which is in the form of an H, which we've also always talked about as an H+. So you're like, well, how's a plus and a plus become negative? This is when you sort of have to say, we're just not even going to think about that part. And then the H pops on the end of there, becoming NAD+, and then it has no charge. And so that's sort of the NADH is sort of the carrier of the H. And so the NAD+ is like the little yellow cart guy, and you're like the hydrogen that gets carried around because you're super important. And so then what happens is the NADH scooches through different levels of membranes and then takes that H+ or that sort of little extra particle down to the electron transport chain. And so whenever we talk about cellular respiration, we talk about photosynthesis, we're talking about building up a gradient. And so we are sort of chunking off different pieces of glucose, taking them, attaching them to a little thing, and then traveling it down and putting it across the membrane. And we're shoving and shoving and shoving hydrogens and hydrogens and hydrogens over until we have a gradient. And so we'll get to that when we get to the electron transport chain in a little bit. And so then in cellular respiration, there's three parts. The first part is glycolysis. It's called glycolysis. But if you break it down, it is the lysis of the glucose. And so it's, if I have glucose, which is C6, and I break it in half, then I have a 3-carbon thing and a 3-carbon thing. And then we have to do something with those. And so that's glycolysis. So that's the first step. When it breaks them down into those 2 3-carbon sugar parts, those are called pyruvates or pyruvic acid. And so then those go into the mitochondria and starts with the citric acid cycle. Sometimes you'll hear it called the Krebs cycle. I almost always call it the citric acid cycle. And then we take those 2 3-carbons and we break them down a little bit more. Every time we break that, we pop off some hydrogens. And those hydrogens get grabbed by the little couriers, and then they get taken down to the other side of the membrane. And then they build up a gradient. And so then we have like a membrane with like things that are like dropping them off, and then like a whole bunch of hydrogens. And then what happens is they build up, they build up, they build up, and then they have to go back across. Excuse me. And so that going back across from on a membrane, so the outside of a mitochondria and the membrane, the inner membranes of a mitochondria, we're sort of in the middle of those. And so we have this built up gradient of hydrogens. And so when those get built up, they come across through a little enzyme called ATP synthase. And so it is the enzyme that synthesizes ATP. And so we have a bunch of ADPs and some inorganic phosphates floating around on one side, and then the zing of the hydrogens through that enzyme comes across, and that energy, the zoom, zing, 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 zing across, pops that that third phosphate onto the ADP and then makes ATP. That's called oxidative phosphorylation. We are moving around hydrogens to phosphorylate, and when we phosphorylate, then we are adding that that phosphorus or that phosphate onto the end of the ADP. So it's sort of taking all that vocabulary and sort of shoving it into a couple of words. And so this is a picture. We're going to draw this, or we're going to label our own. So you'll have a copy of this, and we'll sort of go through the thoughts of that. But this is sort of the overview of cellular respiration. So this is intentionally drawn. The glycolysis is outside of the mitochondria, and it's also could be, you could think of it as fermentation. When glycolysis gets stalled without oxygen, that's fermentation. When it is able to go through the steps, the other steps of cellular respiration, then it gets cellular respiration. So we have different levels of phosphorylation that then gives us different amounts of ATP. And so, but we'll go through this as we go. And so oxidative phosphorylation, so the electron transport chain, the third step in the 3 steps, accounts for almost 90% of the ATP generated. We get 2 ATPs net from glycolysis. We get 2 ATPs net from the citric acid cycle, and then we get about 34 to 36 ATPs with the electron transport chain. So the little bits, the glycolysis, the citric acid cycle, that's called substrate level phosphorylation. And then this just breaks it down. We're going to talk about the little bits and pieces. And so glycolysis is glycolysis. Breaks it down. I talked about this before. And so what happens is you have to spend a little money to make money. And so we have to like exert some energy to break it, to get some energy out. And so overall, we get 4 ATPs, but it took us 2 to do the work. So that's why I said earlier, a net of 2. And then after they get broken in half, and we have 2 3-carbon molecules, so 2 pyruvates or 2 pyruvic acids. Then they go into the mitochondria, and then there's oxygen inside the mitochondria. My lights just on? Maybe? Well, I'll talk in the dark. There we go. And so then we have the pyruvate, and then we have to turn it into what's called acetyl-CoA. And so it is a little intermediate molecule that then is 2 carbons. And so we have 2 3 carbons that come in, and then we sort of lose a carbon from each one as we sort of spin through this little cycle. This carbon, so we take our 3 carbon, we get down to a 2 carbon. This carbon gets linked up with oxygen, and then it gets turned into CO2, and then we breathe it out. And so the CO2 that is produced, that we breathe out, is produced during the citric acid cycle of cellular respiration. And so then this is just showing us a little picture. And then I talked about this before. This takes place inside the mitochondria because that's where oxygen is, and it's necessary for the different things to happen. So as we're going through this little cycle, just like when we broke it in glycolysis and some of the hydrogens came off and they got grabbed by NAD plus and became NADH, and then they sort of traveled along. Now we have that still, and we have another molecule called FADH plus. And so then when we add the H on there, it becomes FADH2. And so then it's sort of the same thing, just a little courier takes it along. And so we have all those things, 6 carbons, all those hydrogens, all those oxygens, and we're sort of breaking them down bit by bit. And we talked a little bit about this earlier, that if we sort of let all the energy that was contained within a molecule of glucose all sort of free at it's just too much. And so we have to sort of bit by bit break it down, take this little hydrogen, put it on this, scooch it over there. And so that's what the FADH and the NAD does for us. And so this is just showing one little cycle through, and then we get some CO2, and we have some sort of byproducts that then really are grabbing the hydrogens and taking them where we need them to be. And so then this is a super complicated process that I don't really ask you to remember, but just know that there's a lot of different steps, there's a lot of different enzymes, and really it comes down to getting NADH and FADH2. We sort of scooch them and we pass them to the other side of the membrane, so we're building up a gradient. We're sort of couriering that hydrogen down to the appropriate part of the mitochondria. And so this just talks about sort of where the money, where we're getting the bang for our buck. And so we get a bunch. This is where we're sort of really getting to the nitty gritty rather than one little break between glucose and we have pyruvate and pyruvate. Now we're sort of going around this little cycle and breaking them off. And so we then we can get a bunch, and they all add up to be about 38. And so then this talks about the different sort of areas of the mitochondria. And so if we think of it like a little kidney bean, and we have this little membrane on the inside, and so then we're passing it over from one side of the little membrane, so in the inside, hopefully, my hand probably don't make sense, but and so we're making a gradient. And so then when they come across back through the ATP synthase, the hydrogen doesn't go onto the ADP and inorganic phosphate. We don't integrate the hydrogen, but we take the zing that it carries across, that then we put into the bond. And so that hydrogen has to go somewhere. And so if you know anything about hydrogens, if you have a hydrogen and that's hydrogen gas, and then we it's like the hydrogen bomb, or like, not hydrogen bomb, but like a balloon filled with hydrogen gas. It's explosive. And so we kind of probably shouldn't have hydrogen gas in our body. So that's the oxygen that we breathe in, comes along, links up to it, and then one of the byproducts is water, which then we use for different cellular processes, and then makes stuff work. And then we have, I talked a little bit about this, and so this is where we have a little enzyme located between the membranes. So we have the gradient on one side of the membrane, we have the sort of oxygen hanging out waiting, some ADP and inorganic phosphate sort of waiting, and then it comes across and it comes through the ATP synthase. that process is also called chemiosmosis. And so it's chemi, like chemistry, like ions, like hydrogens, going from an area of high concentration to an area of low concentration. And so it zings across. And then this is just a little, it didn't look like this in your cell, but sometimes they talk about it works like a little motor. So it sort of goes chick, 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 and every time it pops, makes ATP, 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 ATP. It's water. And so this is just a sort of zoomed in version of the inner and outer membrane, or the sort of sides of the two inner membrane. And so we build up a gradient of hydrogens on one side, and we build that up by dropping them off. The NADH drops it off, and then it becomes charged again, and then the FADH2 drops it off, becomes FAD, FADH plus again, over and over and over and over and over again. And so we're just dropping them off, sending them across. Let's build up a gradient, build up a gradient, build up a gradient, and then it's sort of on the one side of the membrane over there, ready to go. Alrighty, so then this one is just talking about where we get all sort of the ATP, or all the sort of bang for our buck, or when we break all the glucose apart, where does it all go? And so this really, the thing that matters is we're sort of following the hydrogens. And so when we break apart the glucose, that hydrogen, that H plus gets attached to NAD plus. It travels down to the electron transport chain, and then it does like the little motor of chugging through the membrane, and then adding the inorganic phosphate onto the ADP. When we eat food, we really only glean 40% of it. 60% is lost as heat. And then ideally, sort of in perfect conditions, we would get about 38. So 2 from the glycolysis, 2 from the citric acid cycle, and then 34 from the electron transport chain. And so this is just sort of the same picture that we had before, but a little bit different. And so just a little bit more added stuff with the NADH and the FADH2 and all that kind of stuff, but basically the same. And then this talks about when oxygen is not present. And so I talked about this a couple of times, about how we are able to become multicellular and have sort of all of these energy needs because of mitochondria and oxygen. But there are certain things that still function a little bit. And so glycolysis takes place outside of the mitochondria, so glycolysis does not require oxygen, so an anaerobic environment. And then when it does that, when there's no oxygen, then we'll talk about fermentation. And so there's 2 kinds of fermentation that we're going to talk about: alcoholic fermentation and lactic acid fermentation. The difference between the 2 is what kind of organism does them. And so primarily alcohol fermentation occurs in prokaryotes, in yeast. And so it is like if your parent makes like micro brews or whatever, they're pretty big in Colorado, but you're too young for them. Yeast is the thing that they use. Or if you've made bread like from scratch, and then you have to wait for it to like rise, that's the yeast kind of living, and then they produce carbon dioxide, then it makes bubbles. So bubbles in alcohol or bubbles in bread, it's the same thing, it comes from the little yeasties. And then lactic acid fermentation is alrighty, the bell. And so lactic acid occurs in a lot more things, and so we'll talk primarily about us. It also occurs in like some other yeast and fungi, and then some bacteria, but it occurs in animals. And so whenever we do like exercise, and we're like, think if you're like weightlifting and you're doing an activity that you're not really breathing heavy, like running a long distance is aerobic, and so you're breathing in and out. But like if you're sprinting, or if you're like lifting weights, that converts into an anaerobic condition or sort of environment, because you don't breathe heavy. you breathe heavy after, but during, and then as a result, our bodies make lactic acid. And so one of the things that I mentioned almost every unit is sort of how this topic, whatever we're talking about, connects to evolution. And so glycolysis is one of those things. And so it occurs in almost everything, bacteria, animals, prokaryotes, eukaryotes, everything. And so, not everything, but just about everything. And so that is a piece of evidence that if something single-celled way back at the beginning of time, and us super sophisticated, complicated organisms, that we both have that process, the exact same process, leads to sort of the conclusion that we must have come from the same thing. And so evolutionarily, we sort of came from the same sort of cell thing at the beginning of time. And so ATP works like with feedback inhibition. If we need it, we make it. If we don't need it, we sort of hang out. And so we always kind of need it because we're always kind of alive. But if we need extra, then we sort of kick it into gear. And so the key to that is, for us, is breathing. And so if we need more ATP, we breathe heavier. Whether that's from exercise, whether that's from whatever reason, it's our body's sort of mechanism to make us breathe heavier because we need more energy. And you can think of that like exercise. You can think of that like fight or flight. You can think of probably some other examples. And now we'll talk about photosynthesis. And so photosynthesis is just taking light energy and converting it into chemical energy. So we are taking sunshine and we're turning it into glucose. And so this is just some vocab. So autotrophs are producers. They make their own food. So pretty easy to understand. And then photosynthesis occurs in a bunch of different stuff. Plants, algae, some protists, some bacteria. It just depends. They don't necessarily need to be green, but if it's green, it's probably photosynthetic. And so it's a cool little thing that some organisms do. I'd love to be able to photosynthesize, but I cannot. And then heterotrophs is what we are. So we eat something different. So auto means like self and hetero means like different. And so I make my food myself if I'm a plant, and I have to eat something different if I am a heterotroph. And so in like 9th grade or middle school, we called these consumers. And so we are consumers. And so this just talks about chloroplasts and how they are made up. So we'll have some pictures in a bit. Inside a leaf, like in the summertime, they're green. And so inside that leaf, they contain chlorophyll. And chlorophyll is the pigment. Chloroplast is the structure that holds the pigment. And then that enables, that is sort of the pigment that allows photosynthesis to take place. Now, maybe you've learned this in a different class, but maybe you haven't. But the pigments that sort of absorb the energy are not green because the spectra of light, so like Roy G. Biv, is the things that sort of come into it, that light goes into other pigments. The reason that the leaf looks green is because the light is reflected from the green. And so it's sort of like a weird kind of concept. So hopefully that makes sense. And then just structurally, there's little parts on it that have, there's little like holes in the leaf or leaves where gases go in and out of. So it's where the CO2 goes in and the O2 comes out, and they're just called stomata. And then a little bit, a couple of other little words that I just want you to understand is thylakoids is one of them. And so we have in the chloroplast, we have little they look like coins. And so a lot of people describe them as like little stacks of coins. And so those are called thylakoids. And so what that gives us is membranes. And so it gives us a place to like push a gradient across, which sounds a lot like cellular respiration. And structurally, it's similar. And so the sort of concept behind it of chemiosmotic movement, or building up a gradient and bringing it back across a membrane, is very similar between cellular respiration and photosynthesis. And then stroma is just another little structure. We'll look at some pictures and it would probably make more sense. And so now we have a little picture of stomata, and then the mesophyll is sort of the little area, and then down at the bottom we have the thylakoids, which is the little stacks of coins, green coins, and then the whole thing's called a chloroplast, and then inside, sort of what makes these things green, is the chlorophyll. It's just an up-close picture. And then this is the equation. It is the opposite equation from cellular respiration. And so if you can remember one, you can remember the other. I like to remember photosynthesis because for me it's easier. What do plants need? They need sun, they need water, and they need carbon dioxide. And so then they make, what do they make? They make glucose, which is plant structure. Then as a byproduct, they give us oxygen. And then they make a little bit of water. And so if we flip that and substitute ATP for light energy, that's the equation for cellular respiration. And so what happens, sort of the process of molecularly what goes on, is we sort of give water to our plant, we like give it a little drink. And so the sunshine breaks that hydrogen and that oxygen apart. And so then we have some hydrogens. That sounds familiar. And then we have some oxygens, which the plant breathes out. So it's like a waste product for them. But we have like this little hydrogen now. And so then we're going to move that around. And so then this just gives a little detailed, like step-by-step thing, and then we'll look at some pictures. And so a lot of books talk about different words for different things. I like light dependent and light independent. And so some of it depends on light and some of it does not depend on light. Sometimes they call it the Calvin cycle. Sometimes they call it different things. I can't even think of them off the top of my head. But I like light dependent. So the light dependent part is the part that occurs in the sun. And so because we're going to use that sun's energy to do that. And then the light independent is the part that does not require the sun. Not that it works in the darkness, but that it just doesn't need the sun. That's why I like kind of dependent. And then the light reactions, so the ones that must happen in the sunshine, because we need the sunshine to break apart the water. We take that water and we break it into hydrogen and oxygen. We get rid of the oxygen as a waste product, which thank you, plants, because we need it. And then we take NADP plus and we pop that hydrogen on the end and it becomes NADPH. This is the same purpose, slightly different molecular structure than cellular respiration. So we had NADH, NADPH, or I'm sorry, FADH2. And so this is just the one that it's slightly different for photosynthesis. The way that I remember the difference between NADH and NADPH is the one that has P in it is the one that goes with photosynthesis. Both begins with P's. And then it makes ATP from ADP and an inorganic phosphate by photophosphorylation. So the light gives us some energy, and so we can make a little bit of ATP because we're going to do some work, so we need some energy. And so then it goes into a different little cycle or part of the leaf, and then we go through what's called carbon fixation. When you fix something, it's not like repair, it's like a fixative or like to affix, like glue. When you carbon fix, it means you're bringing a carbon in and you're attaching it to the rest of the stuff. And so now we have our hydrogen that came from our water, and now we have carbon dioxide that we're bringing in. And so we have carbon, hydrogen, and oxygen. And so we're going to put them all together and we're going to make glucose. And so if we look at this, and we're going to fill in our little picture in a little bit, but up here on the left-hand side, that sort of light, that's the sunshine. And so it comes in, breaks the oxygen and the hydrogen apart. We get rid of the oxygen. The H goes onto the NADP+, and then we made some little ATP. And then we go into the Calvin cycle, or what I like to call the light independent reactions. And we sort of zing in this little cycle and we bring in some carbon dioxide, we have our hydrogen coming in from the light dependent reactions, and then we produce sugar. And so C6H12O6, that's the sort of simplified formula down at the bottom, but I think you can understand that. And so then at the top, we used energy during that. So we took the ATP that came in at the bottom and we put it into those bonds. And so now we have stored energy. Well, that expends energy from the ATP. So then up at the top, in the sort of middle at the top, it says ADP plus inorganic phosphate. We used up the energy. And so now it's sort of, we have to go get it. We also gave away the hydrogen. And so our NADPH went back to NADP+. And so this just talks about, I talked a little bit about this earlier about the pigments and that absorptions and reflections and different stuff like that. And so then chlorophyll reflects green. And so it appears green, which is a kind of a weird concept. And so this talks about just different ones. And so we have main pigments like chlorophyll, and then we have little bits of chlorophyll, what's called chlorophyll B. And so it's still greenish and it's still used a little bit in photosynthesis, but it's slightly different. And so for the top, it's called chlorophyll A. And then carotenoids are like another kind of auxiliary pigment. And so carotenoids are orange, reddish and orange. And so what happens in the fall is chlorophyll A and chlorophyll B are relatively fragile. And so the cold and the dryness that comes with winter, they break down. And so then we see the other accessory pigments in like fall colors and leaves and pretty trees. But they're there because they help. They're a protective pigment, but they're also pretty, which is nice. And then this is talking about how chloroplasts and mitochondria, just how they compare. They're sort of opposite processes. And so one is we take light and we turn it into chemical. The other one we take chemical and we turn it into mechanical, or we turn it into like energy, ATP that we can use. But it both has membranes, and they both have gradients, and they both have little helper molecules that the little carrier molecules of like NADPH and FADH and all that kind of stuff. And then they have slightly different organelles, so a little bit similar, a little bit different. And just like when I talked about mitochondria sort of being hijacked at the beginning of time, and sort of we took mitochondria in and we were like, yes, make us some energy and we will give you a warm place to live. Chloroplasts have their own DNA as well, which is pretty cool. And so this is just the same stuff that I already talked about, just a little bit more, and sort of how stuff gets pumped across, and same sort of thing, but we make up a gradient and then it zings back through and we can attach it and we can put it onto glucose. And then this just talks about a little bit more about the Calvin cycle, or the light independent reactions. And then this talks a little bit more detail about that light dependent, or the part that does not need the sun, the part where we actually put together the glucose. And so there's little just words I want you to be able to recognize, and then and sort of, I don't expect you to be like a professional like photosynthetic person, but we'll sort of start to process these and maybe be able to recognize them. And so the Calvin cycle, the one big thing is carbon fixation. And so when we get that carbon and the oxygen and the hydrogen, we fix it all together, that's a big deal. And so its enzyme is Rubisco, is what it's called. And then when we talked about, like a week or so ago, we talked about reduction, which means that we are gaining that hydrogen. And so, and then we have the regeneration of the CO2 acceptor, and so we have to, what that gives us is a cycle. And so whenever you see the words regeneration or different steps, oftentimes it has to do with chemical reactions. We talk about cycles. So we talked about the citric acid cycle. We talked about light independent reactions, which is sometimes called the Calvin cycle. They're ones that are going to be some sort of regenerative, and usually it's an enzymatic process that sort of goes zing, 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 and keeps on working and pumping out like products. And so the one was we got like carbon dioxide, and we got some hydrogen off the top, and then we got the oxaloacetate, which you probably don't remember, and that's okay. That's from the citric acid cycle. And then, but this one we sort of zing around, we get glucose, and then it regenerates and regenerates and regenerates. So it makes an efficient, sort of sustainable process. So hopefully that makes sense. And then last little bit is this talks about water and plants, and they have like little protective structures. And so if it is cold or dry, or I'm sorry, hot or dry, the little stomata, the little like where the gases go in and out, the little pores of the plant can close. And so what that does is, if it closes, then the gases can't get in and out. And you might be like, well, that's counterproductive for a plant. So what happens is it slows down the process of photosynthesis, but it also protects the plant from too much water loss. And so it might be like, in the short term, we're in a little bit of trouble, but long term, we're going to survive. And so it's a cool little thing that the plants do that then we can, you know, survive, and then we survive, and everybody survives. Everything is great. So anyway, hopefully that makes sense. Thanks.